Assalamu alaikum again. So let's continue with the second part of the second lecture. So in this lecture we'll talk about a number of things. We'll talk about briefly about light absorbance of nucleic acids. We will talk about the concepts of denaturation of DNA and renaturation, and we will talk about a technique known as dot blot. So in terms of light absorbance of nucleic acid, it just happens that aromatic molecules, chemicals, have the ability to absorb light. And it happens that aromatic pyrimidines and purines can absorb light in the UV range. And the peak absorbance is at 260 nanometers. So this is the wavelength of light that can be absorbed by pyrimidines and purines. And if we do a comparison between single-stranded DNA and double-stranded DNA, in terms of the ability to absorb light, single-stranded DNA has the ability to absorb more light than double-stranded DNA. Okay? So here we have so this plot right here shows light absorbance. So the same concentration in terms of moles, the same concentration of DNA, if it is single-stranded, it absorbs more light than double-stranded DNA. Right. So, and it just happens that if we have double-stranded DNA and the amount is 50 micrograms per mil, it absorbs 1 unit of light at 260 nanometers. So this is absorbance of light at 260 nanometers. So if 1 unit of light is absorbed, it means that this sample has a concentration of DNA of about 50 micrograms per mil. Okay? So here's a question in the bottom right here. What is the concentration of a double-stranded DNA sample diluted at 1 to 10? So I have a sample that is concentrated DNA, and I dilute this sample 1 to 10, meaning that I take one fraction of the DNA sample and I add it to 9 fractions of, let's say, water. Okay, so the DNA sample is diluted. So if the diluted sample absorbs 0.1 units of light, the question is, what is the concentration of the double-stranded DNA sample what is the concentration of the DNA sample, that is the one that is not diluted? Okay, so the idea here is that if I know that 1 unit of light is absorbed by 50 micrograms per mil, so this is how we do it. 0.1 units of light is absorbed times 50 micrograms per 1 unit of light. So unit goes it's eliminated with 0.1 unit with unit, and I'll end up with 0.1 times 50. Well, this means that it's 5. The concentration of the diluted sample is 5 micrograms per mil. Now I want to know the concentration of the sample, the original sample, the sample before dilution. So I multiply this by 10. This is the dilution factor in here. So the concentration of the original DNA sample is 50 micrograms per mil. So I hope this is clear. Okay, so let's move on now to the concept of denaturation. Now, denaturation means that the structure of DNA is disrupted. It's gone. It's not there anymore. So the DNA is not a double helix anymore. And this takes place at high temperature. And if I have, let's say here, a double-stranded DNA at low temperature, at a temperature of about 30 to 50, it is still double-stranded. once I increase the temperature from 50 to 70 to 90, the DNA starts, the hydrogen bonds between the two strands start to be disrupted. Okay? So you can see here that the DNA strands are, they start to be separated from each other. As we increase the temperature, more and more hydrogen bonds are disrupted, and eventually the DNA becomes single-stranded. Now notice here the x-axis is the temperature, and the y-axis is absorbance of DNA. So notice that DNA absorbance increases as it becomes single-stranded, like I mentioned before. Okay? Now, the point of 50% is very important. So this is 1.4, this is 1, the 50% mark is about 1.2. Now the 50% mark is very important in biochemistry, in molecular biology, in pharmacology. So this mark tells us something about how strong the interaction is within this molecule. Okay, so this is the 50% mark, the mark where 50% of the DNA is single-stranded and 50% is double-stranded, is known as the melting temperature. Okay, and this melting temperature differs among different DNA fragments. And so you can imagine that and these are the factors that influence the melting temperature, or TM. The first one is length, and it does make sense. Okay, so the longer the DNA, the higher the melting temperature. In other words, the longer the DNA, the more energy, the more heat is needed to disrupt the hydrogen bonds between the two strands. Okay, so the melting temperature becomes higher. Another factor is the content of G's and C's, okay, or the GC content, the number of base pairs of G's and C's. Remember how I mentioned yesterday that, or in the previous lecture, that the number of hydrogen bonds between G and C is 3, whereas with the A and T it is 2. So if you have a DNA fragment that contains a lot of G's and C's, it means that there are more hydrogen bonds than a DNA fragment of the same length, but it contains more A's and T's. Okay, so this is illustrated here in this plot right here. So you have DNA fragments, you have of the same length, you have one with lower GC content than the other one. Notice that you need more, or you need higher temperature to reach the melting temperature, to reach the 50% mark. Okay, there are other factors that influence the melting temperature, and these include pH. So if you go to the extremes of pH, if you go to very low pH values, or if you place DNA in high pH and alkaline pH, that also disrupts hydrogen bonding. Salts and ions. Well, what do you think? What do you think of the effect of salts and ions on the melting temperature? Would the melting temperature increase or decrease? Well, think about it for a second. Well, remember in the previous lecture how I said that DNA contains, it's a negatively charged molecule, it contains a lot of negative charges because of the phosphate groups, and that causes repulsion between these phosphate groups. So if we add salts and ions, the positively charged ions would mask the positive groups of the DNA. So, what do you think would happen? Well, since negative charges would be masked, that is, they would be hidden as a result of interaction with the positively charged ions, this means that the DNA becomes more stable, and as a result, salts and ions would increase the melting temperature. So the final factor is that I'll talk about is destabilizing agents. There are agents like alkaline solutions, again pH, formamide, as well as urea. What they do is that they disrupt hydrogen bonds, and as a result they lower the melting temperature. Okay, so this is the concept of denaturation of DNA. Now, the opposite of denaturation is known as renaturation. In other words, denaturation is separation of the two strands by breaking up the hydrogen bonds between the two strands. And if we remove the factor that causes denaturation, that is, if we lower the temperature, if we remove the denaturing factors, if we bring pH back to neutral pH, well, that causes renaturation. That is, the two strands would come back together, forming hydrogen bonds to the exact original shape. Why? Because the two strands are complementary to each other. So you will have the binding of the two strands, or the reformation of the original DNA molecule because of that. Okay, so this is the concept of renaturation. Well, hybridization means that it's a hybrid. So we create a hybrid molecule. What does hybrid mean? It means a molecule that is made of two different strands. Okay, so basically if I have DNA strands of different sources, but they are complementary. Let's say that I have human DNA and, I don't know, monkey DNA, and there are, or let's say we have DNA strands from two different human beings, and we separate the two strands from each other by increasing heat, and we lower heat. Well, what would happen? We lower the temperature, that is. What would happen? Well, the two strands would renature again, forming they go back, forming the original the original DNA strands, that is the original DNA, okay, as you can see in here. Or you can have hybrid molecules, that is, you have one strand from one source and another strand from a different source, as long as they are complementary. Okay. Can you have imperfect hybridization? That is, can you have hybridization between two strands that are not exactly complementary to each other? Definitely, you can, as long as you have enough hydrogen bonds between the two strands. You can have formation of hybrids, or imperfect. You can have the formation of imperfect hybridization between two strands. So this is the trick. Okay, so keep that in mind. Now, so let's talk about we'll talk about two hybridization techniques. I'll start in this lecture with a technique known as dot blot. And these two techniques are based on the concept of hybridization. That is the formation of a DNA molecule that is made of two different strands from different sources. And it's based on the concept of complementarity. That is the formation of hydrogen bonds between two strands because they are complementary to each other. Okay, so we can have imperfect hybridization. Let's say that I have a short DNA fragment and a large DNA fragment. Can the small short DNA fragment hybridize with the long DNA fragment? And the answer is definitely yes. Why not? As long as there are enough hydrogen bonds, and as long as the conditions permit the formation of hydrogen bonds, like the ionic strength, the presence of ions in the solution, the temperature, the GC content, and so on, you can have the formation of this hybrid molecule. Well, can imperfect hybridization take place? Yes, again, as long as there are enough hydrogen bonds, you can have imperfect hybridization between a short DNA molecule and a large DNA molecule under uh, uh, permissible conditions. Okay, so let's talk about now these short DNA molecules. These are also known as oligonucleotides. Oligo means short. Okay, so these oligonucleotides, we use them as probes. Probes mean exactly the same thing. So probes are short DNA molecules. They're known as oligonucleotides. The word probe is common. It's a regular English word that means, to probe for something means to search for something. So probes are molecules, are DNA molecules, short DNA molecules that allow us to search if there are complementary sequences within a large DNA molecule to that probe. So the idea here is that we have a probe and we label it. Remember the concept of labeling, DNA labeling, which is basically adding a fluorescent tag or adding a radioactive phosphorus to a probe. So wherever the probe goes, it emits a signal. It gives us a signal telling us that I'm here. Okay? So if I add a probe to a double-stranded DNA, competition would take place between the probe and the complementary strand in forming hydrogen bonds. And since, remember, we're not talking about single probe molecule and single DNA molecule here, we're talking about DNA, millions of DNA molecules of the same type, whether we're talking about DNA strand, large DNA strand, or the short probes or DNA strands, or the oligonucleotides. Okay, so we have these probes in large quantities, and when we add them to a DNA sample, competition would take place between the probe and the complementary strand, this strand right here, and the probe. Since we have larger quantities of probes than the original DNA, the large DNA molecule, the probe would win the competition, and it would form, it would hybridize with the DNA. So if there is interaction between the probe and the DNA, I know that the large DNA molecule contains a sequence that is complementary to the sequence of the probe. Okay? So this is the basis of hybridization techniques. So let's talk about the first technique in here. Okay? So the idea is that I have a large DNA molecule, and it does contain a sequence that is complementary to a probe. Now I know what the sequence of the probe is. Okay, so it's not something that is unknown. And I place my DNA on a piece of paper right here. Okay, and I add a probe, and the probe emits a signal. Well, if there is a sequence that is complementary to the large DNA molecule, then you will have the formation of a interaction, hybridization between the two DNA strands. Okay, now under strict conditions, this interaction does not take place. So I prevent, if there is a difference even in a single base pair, I can prevent this interaction, this hybridization to take place. Now under certain conditions, if I, for example, lower the temperature, if I use a probe that contains a lot of G's and C's, if I play around with the ionic strength of the solution, then I can allow this to happen. But in my case, in the technique that I'll talk about, I prevent this hybridization from taking place between the probe and the large DNA sample. Okay, so let's go over a few concepts in here. This is known as a pedigree. So what you see to the left is known as a pedigree. So it's like a family tree that we use in genetics. So here we have mom and dad, and they have the eye color alleles. So what does an allele mean? Please go back to textbooks, to the net, and if you don't understand what an allele means, so you just look for it yourself, or you can ask me whenever you see me. So basically, genes can exist in different types. So let's say we have the eye color gene, and we all have eye color genes, okay? And these eye color genes exist in different types. So we have the black eye color gene, and we have the blue eye color gene, and we have the brown eye color gene, and so on. And the different types of a gene are known as alleles, okay? So since we are diploid, since we have 2 copies of every chromosome, we can have the same alleles for the eye color gene or different alleles, okay? So let's say that a person would have, this dad has a blue eye color allele and a brown eye color allele. And let's say that the blue allele is dominant. Remember the concept of dominance, سائد ومتنحي. So we have the blue eye color allele that is dominant over the brown eye color allele, okay? So this is imaginary, okay? This is all hypothetical. Now the eye colors, or the color of the eyes of the dad would be blue. Same thing with the mom, okay? She has the blue eye color allele and a brown eye color allele. The blue is dominant over the brown, so the eye color of the mother is also blue. We say that mom and dad are heterozygous for the eye color gene. That is, hetero means different, so they have different alleles for the eye color gene, okay? Now they marry and they have 3 children, and 2 of these children have blue eyes and 1 child has brown eyes, okay? When we look at the alleles of the first child, we find that this child has both alleles being blue, and we say that this child is homozygous for the eye color gene. Homo means identical or the same. Now if we look at the second child, this child also has blue eyes, but if we look at the alleles, this child has blue eye color allele and brown eye color allele, and we said that this child is heterozygous, just like the parents. Now if we look at the third child has brown eyes, and since the brown allele is recessive, متنحي, this means that this child is homozygous for the brown eye color allele or the eye color gene. And indeed, if we look at the genotype, the alleles that this child has, we notice that this child has both alleles being brown, okay? So this is a pedigree. Now we know what the meaning of allele is. It's a type of a gene. And now we know what dominant and recessive mean, as well as homozygous and heterozygous. Okay, so let's go over the technique of dot blot. So this is a technique that tells us if DNA of an individual has a sequence that is complementary to a probe that I use. Remember, I know what the sequence of the probe is. So the idea here is that we take a solid support called a membrane. It's like a piece of paper, and I place the DNA sample on this piece of paper, okay? And I use a probe that is that is tagged or that is labeled with a fluorescent tag, a fluorescent chemical, or radioactive phosphorus. So if I add a probe and the probe binds to the DNA sample, I know that this DNA does have a sequence that is complementary to the probe. If there is no interaction between the probe and the DNA, it means that there is no sequence that is complementary to the probe in the original DNA sample. So how do I know that there's interaction? Because now the spot would give a signal, whether it's radioactive or a fluorescent signal. Okay, so same idea here in the previous slide. I take a sample. They say that I take a sample from different individuals and I place them on this piece of paper. So right here I have individual A, individual B, person C, man D, woman E, whatever. Okay, and I add a probe, and the probe is labeled. Okay, now let's say that this spot right here gives a signal. This means that individual A has in his DNA a sequence that is complementary to the probe. Well, let's say that in this spot right here we have a DNA that does not have a sequence that is complementary to the probe. Then this spot right here would not emit, would not give us a signal. Okay, so that's the idea of a dot blot. Now let's look at this example then. Here we have a family that has a child who has a disease known as cystic fibrosis. Okay, so a disease is known as cystic fibrosis. So here we have a man, square, a woman, circle. They marry, they have 3 children. They have a girl that is affected. She has cystic fibrosis. And they have a boy in square number 2. And this child is not affected. And they have a girl who is not affected as well. Okay, now normally, okay, so cystic fibrosis results from a deletion, that is removal of 3 nucleotides in a specific gene. Okay, so to determine if the family members are homozygous or heterozygous for this particular gene, for that gene that has the deletion, we use what is known as an allele-specific oligonucleotide. Again, we've defined what oligonucleotide is: short DNA molecule, and we use 2 oligonucleotides. We use 2 probes, and they are allele specific. That is, we use an allele specific oligonucleotide that would detect the normal DNA, the DNA that does not have the deletion. And we use another probe that has the ability to detect or to bind, to hybridize with the DNA sequence or the gene or the allele that has the deletion. Okay, so we take DNA from the father, we take DNA from the mother, we take DNA from the 3 children, and we place them on this piece of paper that we call a membrane. So I have the same DNA here of the father, and right here as well. Same thing here for the mother. Okay, same thing here with this girl. We place the DNA here and here. We place the DNA here for the boy and here as well, and we place the DNA for girl number 3 on this piece of on this membrane, and right here and right there. And we add, for the first piece of paper, we add the allele specific nucleotide for the normal DNA sequence that does not have the deletion. We have a signal here, a signal here. There is no signal right here. There's a signal here and a signal right there. What this means is that the father has the normal allele, the mother as well. Now this child does not have the normal allele. This boy has the normal allele, and this girl also has the normal allele. Well, if we add the allele specific oligonucleotide for the sequence that is deleted, that does not have these 3 nucleotides, the AGA. Notice here that for the father's DNA, there is a signal, so the father also has the deleted gene, the mother as well. Child number 1, there is no signal for the DNA of child number 2, and there's a signal for child number 3. This means that the father, the mother, and child number 3, all of them have both the normal allele as well as the defective allele, the allele that has a deletion. And we call them heterozygous since they have 2 different alleles. So hetero means different. So the 3 of them are heterozygous. Now, the boy, the little boy, child number 2, has a signal that comes out if we add the normal oligonucleotide, allele specific oligonucleotide, but not if we use the oligonucleotide that detects the deleted gene. This means that this person has both alleles being normal with no deletion, and we say that this person is homozygous for the normal allele. Now, this little child that has cystic fibrosis, she does not have the normal allele, and there's only a signal coming out if we use the allele specific oligonucleotide or the oligonucleotide for the deleted gene. So we know that she has both alleles having the deletion. She has the defective gene on both chromosomes. And we say that this person is homozygous for the defective gene. She has cystic fibrosis.